Chapter 14. DNA, the genetic material. Concept outline. Section 14.1. What is the genetic material? The Hammerling experiment. Cells store hereditary information in the nucleus. Transplantation experiments. Each cell contains a full set of genetic instructions. The Griffith experiment. Hereditary information can pass between organisms. The Avery and Hershey-Chase experiments. The active principle is DNA. Section 14.2. What is the structure of DNA? The chemical nature of nucleic acids. Nucleic acids are polymers containing 4 nucleotides. The three-dimensional structure of DNA. The DNA molecule is a double helix, with 2 strands held together by base pairing. Section 14.3. How does DNA replicate? The Michelson-Stahl experiment. DNA replication is semi-conservative. The replication process. DNA is replicated by the enzyme DNA polymerase III, working in concert with many other proteins. DNA replicates by assembling a complementary copy of each strand semi-discontinuously. Eukaryotic DNA replication. Eukaryotic chromosomes consist of many zones of replication. Section 14.4. What is a gene? The one gene/one polypeptide hypothesis. A gene encodes all the information needed to express a functional protein or RNA molecule. How DNA encodes protein structure. The nucleotide sequence of a gene dictates the amino acid sequence of the protein it encodes. The realization that patterns of heredity can be explained by the segregation of chromosomes in meiosis raised a question that occupied biologists for over 50 years. What is the exact nature of the connection between hereditary traits and chromosomes? This chapter describes the chain of experiments that have led to our current understanding of the molecular mechanisms of heredity, Figure 14.1. The experiments are among the most elegant in science. Just as in a good detective story, each conclusion has led to new questions. The intellectual path taken has not always been a straight one, the best questions not always obvious. But however erratic and lurching the course of the experimental journey, our picture of heredity has become progressively clearer, the image more sharply defined. Section 14.1. What is the genetic material? The Hammerling experiment. Cells store hereditary information in the nucleus. Perhaps the most basic question one can ask about hereditary information is where it is stored in the cell. To answer this question, Danish biologist Joachim Hammerling, working at the Max Planck Institute for Marine Biology in Berlin in the 1930s, cut cells into pieces and observed the pieces to see which were able to express hereditary information. For this experiment, Hammerling needed cells large enough to operate on conveniently and differentiated enough to distinguish the pieces. He chose the unicellular green alga acetabularia, which grows up to 5 centimeters, as a model organism for his investigations. Just as Mendel used pea plants and Sturtevant used fruit flies as model organisms, Hammerling picked an organism that was suited to the specific experimental question he wanted to answer, assuming that what he learned could then be applied to other organisms. Individuals of the genus Acetabularia have distinct foot, stalk, and cap regions, all are differentiated parts of a single cell. The nucleus is located in the foot. As a preliminary experiment, Hammerling amputated the caps of some cells and the feet of others. He found that when he amputated the cap, a new cap regenerated from the remaining portions of the cell, foot and stalk. When he amputated the foot, however, no new foot regenerated from the cap and stalk. Hammerling, therefore, hypothesized that the hereditary information resided within the foot of Acetabularia. Surgery on single cells. To test his hypothesis, Hammerling selected individuals from 2 species of the genus Acetabularia in which the caps look very different from one another. Mediterranea has a disc-shaped cap, and Crenulata has a branched, flower-like cap. Hammerling grafted a stalk from Crenulata to a foot from Mediterranea, Figure 14.2. The cap that regenerated looked somewhat like the cap of a Crenulata, though not exactly the same. Hammerling then cut off this regenerated cap and found that a disc-shaped cap exactly like that of a metatracheia formed in the second regeneration and in every regeneration thereafter. This experiment supported Hammerling's hypothesis that the instructions specifying the kind of cap are stored in the foot of the cell, and that these instructions must pass from the foot through the stalk to the cap. In this experiment, the initial flower-shaped cap was somewhat intermediate in shape, unlike the disc-shaped caps of subsequent generations. Hammerling speculated that this initial cap, which resembled that of a crenulata, was formed from instructions already present in the transplanted stalk when it was excised from the original A. crenulata cell. In contrast, all of the caps that regenerated subsequently used new information derived from the foot of the A. metatracheia cell the stalk had been grafted onto. In some unknown way, the original instructions that had been present in the stalk were eventually used up. We now understand that genetic instructions, in the form of messenger RNA, discussed in Chapter 15, pass from the nucleus in the foot upward through the stalk to the developing cap. Synopsis. Hereditary information in acetabularia is stored in the foot of the cell, where the nucleus resides. Transplantation experiments, each cell contains a full set of genetic instructions. Because the nucleus is contained in the foot of acetabularia, Hammerling's experiments suggested that the nucleus is the repository of hereditary information in a cell. A direct test of this hypothesis was carried out in 1952 by American embryologists Robert Briggs and Thomas King. Using a glass pipette drawn to a fine tip and working with a microscope, Briggs and King removed the nucleus from a frog egg. Without the nucleus, the egg did not develop. However, when they replaced the nucleus with one removed from a more advanced frog embryo cell, the egg developed into an adult frog. Clearly, the nucleus was directing the egg's development, figure 14.3. Successfully transplanting nuclei. Can every nucleus in an organism direct the development of an entire adult individual? The experiment of Briggs and King did not answer this question definitively, because the nuclei they transplanted from frog embryos into eggs often caused the eggs to develop abnormally. Two experiments performed soon afterward gave a clearer answer to the question. In the first, John Gordon, working with another species of frog at Oxford and Yale, transplanted nuclei from tadpole cells into eggs from which the nuclei had been removed. The experiments were difficult. It was necessary to synchronize the division cycles of donor and host. However, in many experiments, the eggs went on to develop normally, indicating that the nuclei of cells in later stages of development retain the genetic information necessary to direct the development of all other cells in an individual. Totipotency in plants. In the second experiment, F. C. Stewart at Cornell University in 1958 placed small fragments of fully developed carrot tissue, isolated from a part of the vascular system called the phloem, in a flask containing liquid growth medium. Stewart observed that when individual cells broke away from the fragments, they often divided and developed into multicellular roots. When he immobilized the roots by placing them in a solid growth medium, they went on to develop normally into entire, mature plants. Stewart's experiment makes it clear that, even in adult tissues, the nuclei of individual plant cells are totipotent, each contains a full set of hereditary instructions and can generate an entire adult individual. As you will learn in Chapter 19, animal cells, like plant cells, can be totipotent, and a single adult animal cell can generate an entire adult animal. Synopsis. Hereditary information is stored in the nucleus of eukaryotic cells. Each nucleus in any eukaryotic cell contains a full set of genetic instructions. The Griffith experiment, hereditary information can pass between organisms. The identification of the nucleus as the repository of hereditary information focused attention on the chromosomes, which were already suspected to be the vehicles of Mendelian inheritance. Specifically, biologists wondered how the genes, the units of hereditary information studied by Mendel, were actually arranged in the chromosomes. They knew that chromosomes contained both protein and deoxyribonucleic acid, DNA. Which of these held the genes? Starting in the late 1920s and continuing for about 30 years, a series of investigations addressed this question. In 1928, British microbiologist Frederick Griffith made a series of unexpected observations while experimenting with pathogenic, disease-causing, bacteria. When he infected mice with a virulent strain of Streptococcus pneumoniae bacteria, then known as pneumococcus, the mice died of blood poisoning. However, when he infected similar mice with a mutant strain of S. pneumoniae that lacked the virulent strain's polysaccharide coat, the mice showed no ill effects. The coat was apparently necessary for virulence. The normal pathogenic form of this bacterium is referred to as the S form because it forms smooth colonies on a culture dish. The mutant form, which lacks an enzyme needed to manufacture the polysaccharide capsule, is called the R form because it forms rough colonies. To determine whether the polysaccharide coat itself had a toxic effect, Griffith injected dead bacteria of the virulent S strain into mice. The mice remained perfectly healthy. As a control, he injected mice with a mixture containing dead S bacteria of the virulent strain and live coatless R bacteria, each of which by itself did not harm the mice, figure 14.4. Unexpectedly, the mice developed disease symptoms and many of them died. The blood of the dead mice was found to contain high levels of live, virulent Streptococcus type S bacteria, which had surface proteins characteristic of the live, previously R, strain. Somehow, the information specifying the polysaccharide coat had passed from the dead, virulent S bacteria to the live, coatless R bacteria in the mixture, permanently transforming the coatless R bacteria into the virulent S variety. Transformation is the transfer of genetic material from one cell to another and can alter the genetic makeup of the recipient cell. Synopsis. Hereditary information can pass from dead cells to living ones, transforming them. The Avery and Hershey-Chase experiments, the active principle is DNA. The Avery experiments. The agent responsible for transforming Streptococcus went undiscovered until 1944. In a classic series of experiments, Oswald Avery and his co-workers Colin MacLeod and Macklin McCarty characterized what they referred to as the transforming principle. They first prepared the mixture of dead S. Streptococcus and live R. Streptococcus that Griffith had used. Then Avery and his colleagues removed as much of the protein as they could from their preparation, eventually achieving 99.98% purity. Despite the removal of nearly all protein, the transforming activity was not reduced. Moreover, the properties of the transforming principle resembled those of DNA in several ways. 1. When the purified principle was analyzed chemically, the array of elements agreed closely with DNA. 2. When spun at high speeds in an ultracentrifuge, the transforming principle migrated to the same level, density, as DNA. 3. Extracting the lipid and protein from the purified transforming principle did not reduce its activity. 4. Protein-digesting enzymes did not affect the principle's activity, nor did RNA-digesting enzymes. 5. The DNA-digesting enzyme DNase destroyed all transforming activity. The evidence was overwhelming. They concluded that a nucleic acid of the deoxyribose type is the fundamental unit of the transforming principle of pneumococcus type 3, in essence, that DNA is the hereditary material. The Hershey-Chase experiment. Avery's results were not widely accepted at first, as many biologists preferred to believe that proteins were the repository of hereditary information. Additional evidence supporting Avery's conclusion was provided in 1952 by Alfred Hershey and Martha Chase, who experimented with bacteriophages, viruses that attack bacteria. Viruses, described in more detail in Chapter 33, consist of either DNA or RNA, ribonucleic acid, surrounded by a protein coat. When a lytic, potentially cell-rupturing, bacteriophage infects a bacterial cell, it first binds to the cell's outer surface and then injects its hereditary information into the cell. There, the hereditary information directs the production of thousands of new viruses within the bacterium. The bacterial cell eventually ruptures, or lyses, releasing the newly made viruses. To identify the hereditary material injected into bacterial cells at the start of an infection, Hershey and Chase used the bacteriophage T2, which contains DNA rather than RNA. They labeled the two parts of the viruses, the DNA and the protein coat, with different radioactive isotopes that would serve as tracers. In some experiments, the viruses were grown on a medium containing an isotope of phosphorus, 32P, and the isotope was incorporated into the phosphate groups of newly synthesized DNA molecules. In other experiments, the viruses were grown on a medium containing 35S, an isotope of sulfur, which is incorporated into the amino acids of newly synthesized protein coats. The 32P and 35S isotopes are easily distinguished from each other because they emit particles with different energies when they decay. After the labeled viruses were permitted to infect bacteria, the bacterial cells were agitated violently to remove the protein coats of the infecting viruses from the surfaces of the bacteria. This procedure removed nearly all of the 35S label, and thus nearly all of the viral protein, from the bacteria. However, the 32P label, and thus the viral DNA, had transferred to the interior of the bacteria, figure 14.5, and was found in viruses subsequently released from the infected bacteria. Hence, the hereditary information injected into the bacteria that specified the new generation of viruses was DNA and not protein. Synopsis. Avery's experiments demonstrate conclusively that DNA is Griffith's transforming material. The hereditary material of bacteriophages is DNA and not protein. Section 14.2. What is the structure of DNA? The chemical nature of nucleic acids. A German chemist, Friedrich Miescher, discovered DNA in 1869, only 4 years after Mendel's work was published. Miescher extracted a white substance from the nuclei of human cells and fish sperm. The proportion of nitrogen and phosphorus in the substance was different from that in any other known constituent of cells, which convinced Miescher that he had discovered a new biological substance. He called this substance nuclein, because it seemed to be specifically associated with the nucleus. Levine's analysis, DNA is a polymer. Because Miescher's nuclein was slightly acidic, it came to be called nucleic acid. For 50 years biologists did little research on the substance, because nothing was known of its function in cells. In the 1920s, the basic structure of nucleic acids was determined by the biochemist P. A. Levine, who found that DNA contains 3 main components, figure 14.6, 1, phosphate, PO4, groups, 2, 5-carbon sugars, and 3, nitrogen-containing bases called purines, adenine, A, and guanine, G, and pyrimidines, thymine, T, and cytosine, C. RNA contains uracil, U, instead of T. From the roughly equal proportions of these components, Levine concluded correctly that DNA and RNA molecules are made of repeating units of the 3 components. Each unit, consisting of a sugar attached to a phosphate group and a base, is called a nucleotide. The identity of the base distinguishes one nucleotide from another. To identify the various chemical groups in DNA and RNA, it is customary to number the carbon atoms of the base and the sugar and then refer to any chemical group attached to a carbon atom by that number. In the sugar, 4 of the carbon atoms together with an oxygen atom form a 5-membered ring. As illustrated in figure 14.7, the carbon atoms are numbered 1' to 5', proceeding clockwise from the oxygen atom. The prime symbol indicates that the number refers to a carbon in a sugar rather than a base. Under this numbering scheme, the phosphate group is attached to the 5' carbon atom of the sugar, and the base is attached to the 1' carbon atom. In addition, a free hydroxyl, OH, group is attached to the 3' carbon atom. The 5' phosphate and 3' hydroxyl groups allow DNA and RNA to form long chains of nucleotides, because these 2 groups can react chemically with each other. The reaction between the phosphate group of one nucleotide and the hydroxyl group of another is a dehydration synthesis, eliminating a water molecule and forming a covalent bond that links the 2 groups, figure 14.8. The linkage is called a phosphodiester bond because the phosphate group is now linked to the 2 sugars by means of a pair of ester, cosine bonds. The 2-unit polymer resulting from this reaction still has a free 5' phosphate group at one end and a free 3' hydroxyl group at the other, so it can link to other nucleotides. In this way, many thousands of nucleotides can join together in long chains. Linear strands of DNA or RNA, no matter how long, will almost always have a free 5' phosphate group at one end and a free 3' hydroxyl group at the other. Therefore, every DNA and RNA molecule has an intrinsic directionality, and we can refer unambiguously to each end of the molecule. By convention, the sequence of bases is usually expressed in the 5' to 3' direction. Thus, the base sequence GTCCAT refers to the sequence 5' PGPTPCPCPAPT, OH3', where the phosphates are indicated by P. Note that this is not the same molecule as that represented by the reverse sequence 5' PTPAPCPCPTPG, OH3'. Levine's early studies indicated that all 4 types of DNA nucleotides were present in roughly equal amounts. This result, which later proved to be erroneous, led to the mistaken idea that DNA was a simple polymer in which the 4 nucleotides merely repeated, for instance, GCAT. 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 If the sequence never varied, it was difficult to see how DNA might contain the hereditary information. This was why Avery's conclusion that DNA is the transforming principle was not readily accepted at first. It seemed more plausible that DNA was simply a structural element of the chromosomes, with proteins playing the central genetic role. Chargaff's analysis: DNA is not a simple repeating polymer. When Levine's chemical analysis of DNA was repeated using more sensitive techniques that became available after World War II, quite a different result was obtained. The 4 nucleotides were not present in equal proportions in DNA molecules after all. A careful study carried out by Erwin Chargaff showed that the nucleotide composition of DNA molecules varied in complex ways, depending on the source of the DNA, Table 14.1. This strongly suggested that DNA was not a simple repeating polymer and might have the information encoding properties genetic material must have. Despite DNA's complexity, however, Chargaff observed an important underlying regularity in double-stranded DNA. The amount of adenine present in DNA always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. These findings are commonly referred to as Chargaff's rules. 1. The proportion of A always equals that of T, and the proportion of G always equals that of C, A equals T, and G equals C. 2. It follows that there is always an equal proportion of purines, A and G, and pyrimidines, C and T. Synopsis: a single strand of DNA or RNA consists of a series of nucleotides joined together in a long chain. In all natural double-stranded DNA molecules, the proportion of A equals that of T, and the proportion of G equals that of C. The three-dimensional structure of DNA. As it became clear that DNA was the molecule that stored the hereditary information, investigators began to puzzle over how such a seemingly simple molecule could carry out such a complex function. Franklin, X-ray diffraction patterns of DNA. The significance of the regularities pointed out by Chargaff were not immediately obvious, but they became clear when a British chemist, Rosalind Franklin, figure 14.9a, carried out an X-ray diffraction analysis of DNA. In X-ray diffraction, a molecule is bombarded with a beam of X-rays. When individual rays encounter atoms, their path is bent or diffracted, and the diffraction pattern is recorded on photographic film. The patterns resemble the ripples created by tossing a rock into a smooth lake, figure 14.9b. When carefully analyzed, they yield information about the three-dimensional structure of a molecule. X-ray diffraction works best on substances that can be prepared as perfectly regular crystalline arrays. However, it was impossible to obtain true crystals of natural DNA at the time Franklin conducted her analysis, so she had to use DNA in the form of fibers. Franklin worked in the laboratory of British biochemist Maurice Wilkins, who was able to prepare more uniformly oriented DNA fibers than anyone had previously. Using these fibers, Franklin succeeded in obtaining crude diffraction information on natural DNA. The diffraction patterns she obtained suggested that the DNA molecule had the shape of a helix, or corkscrew, with a diameter of about 2 nanometers and a complete helical turn every 3.4 nanometers, figure 14.9c. Watson and Crick, a model of the double helix. Learning informally of Franklin's results before they were published in 1953, James Watson and Francis Crick, two young investigators at Cambridge University, quickly worked out a likely structure for the DNA molecule, figure 14.10, which we now know was substantially correct. They analyzed the problem deductively, first building models of the nucleotides, and then trying to assemble the nucleotides into a molecule that matched what was known about the structure of DNA. They tried various possibilities before they finally hit on the idea that the molecule might be a simple double helix, with the bases of two strands pointed inward toward each other, forming base pairs. In their model, base pairs always consist of purines, which are large, pointing toward pyrimidines, which are small, keeping the diameter of the molecule a constant 2 nanometers. Because hydrogen bonds can form between the bases in a base pair, the double helix is stabilized as a duplex DNA molecule composed of two antiparallel strands, one chain running 3' to 5' and the other 5' to 3'. The base pairs are planar, flat, and stack 0.34 nanometers apart as a result of hydrophobic interactions, contributing to the overall stability of the molecule. The Watson-Crick model explained why Chargaff had obtained the results he had. In a double helix, adenine forms two hydrogen bonds with thymine, but it will not form hydrogen bonds properly with cytosine. Similarly, guanine forms 3 hydrogen bonds with cytosine, but it will not form hydrogen bonds properly with thymine. Consequently, adenine and thymine will always occur in the same proportions in any DNA molecule, as will guanine and cytosine, because of this base pairing. Synopsis. The DNA molecule is a double helix, the strands held together by base pairing. Section 14.3. How does DNA replicate? The Michelson-Stahl experiment, DNA replication is semi-conservative. The Watson-Crick model immediately suggested that the basis for copying the genetic information is complementarity. One chain of the DNA molecule may have any conceivable base sequence, but this sequence completely determines the sequence of its partner in the duplex. For example, if the sequence of one chain is 5' @cat 3', the sequence of its partner must be 3' TAACGTA 5'. Thus, each chain in the duplex is a complement of the other. The complementarity of the DNA duplex provides a ready means of accurately duplicating the molecule. If one were to unzip the molecule, one would need only to assemble the appropriate complementary nucleotides on the exposed single strands to form two daughter duplexes with the same sequence. This form of DNA replication is called semi-conservative, because while the sequence of the original duplex is conserved after one round of replication, the duplex itself is not. Instead, each strand of the duplex becomes part of another duplex. Two other hypotheses of gene replication were also proposed. The conservative model stated that the parental double helix would remain intact and generate DNA copies consisting of entirely new molecules. The dispersive model predicted that parental DNA would become dispersed throughout the new copy so that each strand of all the daughter molecules would be a mixture of old and new DNA. The three hypotheses of DNA replication were evaluated in 1958 by Matthew Meselson and Franklin Stahl of the California Institute of Technology. These two scientists grew bacteria in a medium containing the heavy isotope of nitrogen, 15N, which became incorporated into the bases of the bacterial DNA. After several generations, the DNA of these bacteria was denser than that of bacteria grown in a medium containing the lighter isotope of nitrogen, 14N. Meselson and Stahl then transferred the bacteria from the 15N medium to the 14N medium and collected the DNA at various intervals. By dissolving the DNA they had collected in a heavy salt called cesium chloride and then spinning the solution at very high speeds in an ultracentrifuge, Michelson and Stahl were able to separate DNA strands of different densities. The enormous centrifugal forces generated by the ultracentrifuge caused the cesium ions to migrate toward the bottom of the centrifuge tube, creating a gradient of cesium concentration, and thus of density. Each DNA strand floats or sinks in the gradient until it reaches the position where its density exactly matches the density of the cesium there. Because 15N strands are denser than 14N strands, they migrate farther down the tube to a denser region of the cesium gradient. The DNA collected immediately after the transfer was all dense. However, after the bacteria completed their first round of DNA replication in the 14N medium, the density of their DNA had decreased to a value intermediate between 14N DNA and 15N DNA. After the second round of replication, two density classes of DNA were observed, one intermediate and one equal to that of 14N DNA, figure 14.11. Michelson and Stahl interpreted their results as follows. After the first round of replication, each daughter DNA duplex was a hybrid possessing one of the heavy strands of the parent molecule and one light strand. When this hybrid duplex replicated, it contributed one heavy strand to form another hybrid duplex and one light strand to form a light duplex, figure 14.12. Thus, this experiment clearly confirmed the prediction of the Watson-Crick model that DNA replicates in a semi-conservative manner. Synopsis. The basis for the great accuracy of DNA replication is complementarity. A DNA molecule is a duplex, containing two strands that are complementary mirror images of each other, so either one can be used as a template to reconstruct the other. The replication process. To be effective, DNA replication must be fast and accurate. The machinery responsible has been the subject of intensive study for 40 years, and we now know a great deal about it. The replication of DNA begins at one or more sites on the DNA molecule where there is a specific sequence of nucleotides called a replication origin, figure 14.13. There the DNA replicating enzyme DNA polymerase III and other enzymes begin a complex process that catalyzes the addition of nucleotides to the growing complementary strands of DNA, figure 14.14. Table 14.2 lists the proteins involved in DNA replication in bacteria. Before considering the replication process in detail, let's take a closer look at DNA polymerase III. DNA polymerase III. The first DNA polymerase enzyme to be characterized, DNA polymerase I of the bacterium Escherichia coli, is a relatively small enzyme that plays a key supporting role in DNA replication. The true E. coli replicating enzyme, dubbed DNA polymerase III, is some 10 times larger and far more complex in structure. We know more about DNA polymerase III than any other organism's DNA polymerase, and so we'll describe it in detail here. Other DNA polymerases are thought to be broadly similar. DNA polymerase III contains 10 different kinds of polypeptide chains, as illustrated in figure 14.15. The enzyme is a dimer, with two similar multisubunit complexes. Each complex catalyzes the replication of one DNA strand. A variety of different proteins play key roles within each complex. The subunits include a single large catalytic alpha subunit that catalyzes 5' to 3' addition of nucleotides to a growing chain, a smaller epsilon subunit that proofreads 3' to 5' for mistakes, and a ring-shaped beta-2 dimer subunit that clamps the polymerase III complex around the DNA double helix. Polymerase III progressively threads the DNA through the enzyme complex, moving it at a rapid rate, some 1,000 nucleotides per second, 100 full turns of the helix, 0.34 micrometers. The need for a primer. One of the features of DNA polymerase III is that it can add nucleotides only to a chain of nucleotides that is already paired with the parent strands. Hence, DNA polymerase cannot link the first nucleotides in a newly synthesized strand. Instead, another enzyme, an RNA polymerase called primase, constructs an RNA primer, a sequence of about 10 RNA nucleotides complementary to the parent DNA template. DNA polymerase III recognizes the primer and adds DNA nucleotides to it to construct the new DNA strands. The RNA nucleotides in the primers are then replaced by DNA nucleotides. The two strands of DNA are assembled in different ways. Another feature of DNA polymerase III is that it can add nucleotides only to the 3' end of a DNA strand, the end with an OH group attached to a 3' carbon atom. This means that replication always proceeds in the 5' to 3' direction on a growing DNA strand. Because the two parent strands of a DNA molecule are antiparallel, the new strands are oriented in opposite directions along the parent templates at each replication fork, figure 14.16. Therefore, the new strands must be elongated by different mechanisms. The leading strand, which elongates toward the replication fork, is built up simply by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments that are later connected. These segments, called Okazaki fragments, are about 100 to 200 nucleotides long in eukaryotes and 1000 to 2000 nucleotides long in prokaryotes. Each Okazaki fragment is synthesized by DNA polymerase III in the 5' to 3' direction, beginning at the replication fork and moving away from it. When the polymerase reaches the 5' end of the lagging strand, another enzyme, DNA ligase, attaches the fragment to the lagging strand. The DNA is further unwound, new RNA primers are constructed, and DNA polymerase III then jumps ahead 1000 to 2000 nucleotides, toward the replication fork, to begin constructing another Okazaki fragment. If one looks carefully at electron micrographs showing DNA replication in progress, one can sometimes see that one of the parent strands near the replication fork appears single-stranded over a distance of about 1000 nucleotides. Because the synthesis of the leading strand is continuous, while that of the lagging strand is discontinuous, the overall replication of DNA is said to be semi-discontinuous. The replication process. The replication of the DNA double helix is a complex process that has taken decades of research to understand. It takes place in 5 interlocking steps. 1. Opening up the DNA double helix. The very stable DNA double helix must be opened up and its strands separated from each other for semi-conservative replication to occur. Stage 1. Initiating replication. The binding of initiator proteins to the replication origin starts an intricate series of interactions that opens the helix. Stage 2. Unwinding the duplex. After initiation, unwinding enzymes called helicases bind to and move along one strand, shouldering aside the other strand as they go. Stage 3. Stabilizing the single strands. The unwound portion of the DNA double helix is stabilized by single-strand binding protein, which binds to the exposed single strands, protecting them from cleavage and preventing them from rewinding. Stage 4. Relieving the torque generated by unwinding. For replication to proceed at 1000 nucleotides per second, the parental helix ahead of the replication fork must rotate 100 revolutions per second. To relieve the resulting twisting, called torque, enzymes known as toposomerases, or, more informally, gyrases, cleave a strand of the helix, allow it to swivel around the intact strand, and then reseal the broken strand. 2. Building a primer. New DNA cannot be synthesized on the exposed templates until a primer is constructed, as DNA polymerases require 3-foot primers to initiate replication. The necessary primer is a short stretch of RNA, added by a specialized RNA polymerase called primase in a multi-subunit complex informally called a primosome. Why an RNA primer, rather than DNA? Starting chains on exposed templates introduces many errors. RNA marks this initial stretch as temporary, making this error-prone stretch easy to excise later. 3. Assembling complementary strands. Next, the dimeric DNA polymerase III then binds to the replication fork. While the leading strand complexes with 1/2 of the polymerase dimer, the lagging strand is thought to loop around and complex with the other half of the polymerase dimer, figure 14.17. Moving in concert down the parental double helix, DNA polymerase III catalyzes the formation of complementary sequences on each of the two single strands at the same time. 4. Removing the primer. The enzyme DNA polymerase I now removes the RNA primer and fills in the gap, as well as any gaps between Okazaki fragments. 5. Joining the Okazaki fragments. After any gaps between Okazaki fragments are filled in, the enzyme DNA ligase joins the fragments to the lagging strand. Synopsis. DNA replication involves many different proteins that open and unwind the DNA double helix, stabilize the single strands, synthesize RNA primers, assemble new complementary strands on each exposed parental strand, one of them discontinuously, remove the RNA primer, and join new discontinuous segments on the lagging strand. Eukaryotic DNA replication. In eukaryotic cells, the DNA is packaged in nucleosomes within chromosomes, figure 14.18. Each individual zone of a chromosome replicates as a discrete section called a replication unit, or replicon, which can vary in length from 10,000 to 1 million base pairs, most are about 100,000 base pairs long. Each replication unit has its own origin of replication, and multiple units may be undergoing replication at any given time, as can be seen in electron micrographs of replicating chromosomes, figure 14.19. Each unit replicates in a way fundamentally similar to prokaryotic DNA replication, using similar enzymes. The advantage of having multiple origins of replication in eukaryotes is speed. Replication takes approximately 8 hours in human cells, but if there were only one origin, it would take 100 times longer. Regulation of the replication process ensures that only one copy of the DNA is ultimately produced. How a cell achieves this regulation is not yet completely clear. It may involve periodic inhibitor or initiator proteins on the DNA molecule itself. Synopsis. Eukaryotic chromosomes have multiple origins of replication. Section 14.4. What is a gene? The one gene/one polypeptide hypothesis. As the structure of DNA was being solved, other biologists continued to puzzle over how the genes of Mendel were related to DNA. Garrod, inherited disorders can involve specific enzymes. In 1902, a British physician, Archibald Garrod, was working with one of the early Mendelian geneticists, his countryman William Bateson, when he noted that certain diseases he encountered among his patients seemed to be more prevalent in particular families. By examining several generations of these families, he found that some of the diseases behaved as if they were the product of simple recessive alleles. Garrod concluded that these disorders were Mendelian traits and that they had resulted from changes in the hereditary information in an ancestor of the affected families. Garrod investigated several of these disorders in detail. In alkaptonuria, the patients produced urine that contained homogentisic acid, alkapton. This substance oxidized rapidly when exposed to air, turning the urine black. In normal individuals, homogentisic acid is broken down into simpler substances. With considerable insight, Garrod concluded that patients suffering from alkaptonuria lacked the enzyme necessary to catalyze this breakdown. He speculated that many other inherited diseases might also reflect enzyme deficiencies. Beadle and Tatum, genes specify enzymes. From Garrod's finding, it took but a short leap of intuition to surmise that the information encoded within the DNA of chromosomes acts to specify particular enzymes. This point was not actually established, however, until 1941, when a series of experiments by Stanford University geneticists George Beadle and Edward Tatum provided definitive evidence on this point. Beadle and Tatum deliberately set out to create Mendelian mutations in chromosomes and then studied the effects of these mutations on the organism, figure 14.20. A defined system. One of the reasons Beadle and Tatum's experiments produced clear-cut results is that the researchers made an excellent choice of experimental organism. They chose the bread mold Neurospora, a fungus that can be grown readily in the laboratory on a defined medium, a medium that contains only known substances such as glucose and sodium chloride, rather than some uncharacterized mixture of substances such as ground-up yeasts. Beadle and Tatum exposed neurospora spores to X-rays, expecting that the DNA in some of the spores would experience damage in regions encoding the ability to make compounds needed for normal growth, see Figure 14.20. DNA changes of this kind are called mutations, and organisms that have undergone such changes, in this case losing the ability to synthesize one or more compounds, are called mutants. Initially, they allowed the progeny of the irradiated spores to grow on a defined medium containing all of the nutrients necessary for growth, so that any growth-deficient mutants resulting from the irradiation would be kept alive. Isolating growth-deficient mutants. To determine whether any of the progeny of the irradiated spores had mutations causing metabolic deficiencies, Beadle and Tatum placed subcultures of individual fungal cells on a minimal medium that contained only sugar, ammonia, salts, a few vitamins, and water. Cells that had lost the ability to make other compounds necessary for growth would not survive on such a medium. Using this approach, Beadle and Tatum succeeded in identifying and isolating many growth-deficient mutants. Identifying the deficiencies. Next, the researchers added various chemicals to the minimal medium in an attempt to find one that would enable a given mutant strain to grow. This procedure allowed them to pinpoint the nature of the biochemical deficiency that strain had. The addition of arginine, for example, permitted several mutant strains, dubbed ARG mutants, to grow. When their chromosomal positions were located, the ARG mutations were found to cluster in 3 areas, Figure 14.21. 1 gene/1 polypeptide. For each enzyme in the arginine biosynthetic pathway, Beadle and Tatum were able to isolate a mutant strain with a defective form of that enzyme, and the mutation was always located at one of a few specific chromosomal sites. Most importantly, they found there was a different site for each enzyme. Thus, each of the mutants they examined had a defect in a single enzyme, caused by a mutation at a single site on one chromosome. Beadle and Tatum concluded that genes produce their effects by specifying the structure of enzymes and that each gene encodes the structure of one enzyme. They called this relationship the one gene/one enzyme hypothesis. Because many enzymes contain multiple protein or polypeptide subunits, each encoded by a separate gene, the relationship is today more commonly referred to as one gene/one polypeptide. Enzymes are responsible for catalyzing the synthesis of all the parts of an organism. They mediate the assembly of nucleic acids, proteins, carbohydrates, and lipids. Therefore, by encoding the structure of enzymes and other proteins, DNA specifies the structure of the organism itself. Synopsis. Genetic traits are expressed largely as a result of the activities of enzymes. Organisms store hereditary information by encoding the structures of enzymes and other proteins in their DNA. How DNA encodes protein structure. What kind of information must a gene encode to specify a protein? For some time, the answer to that question was not clear, as protein structure seemed impossibly complex. Sanger, proteins consist of defined sequences of amino acids. The picture changed in 1953, the same year in which Watson and Crick unraveled the structure of DNA. That year, the English biochemist Frederick Sanger, after many years of work, announced the complete sequence of amino acids in the protein insulin. Insulin, a small protein hormone, was the first protein for which the amino acid sequence was determined. Sanger's achievement was extremely significant because it demonstrated for the first time that proteins consisted of definable sequences of amino acids. For any given form of insulin, every molecule has the same amino acid sequence. Sanger's work soon led to the sequencing of many other proteins, and it became clear that all enzymes and other proteins are strings of amino acids arranged in a certain definite order. The information needed to specify a protein such as an enzyme, therefore, is an ordered list of amino acids. Ingram, single amino acid changes in a protein can have profound effects. Following Sanger's pioneering work, Vernon Ingram in 1956 discovered the molecular basis of sickle cell anemia, a protein defect inherited as a Mendelian disorder. By analyzing the structures of normal and sickle cell hemoglobin, Ingram, working at Cambridge University, showed that sickle cell anemia is caused by a change from glutamic acid to valine at a single position in the protein, figure 14.22. The alleles of the gene encoding hemoglobin differed only in their specification of this one amino acid in the hemoglobin amino acid chain. These experiments and other related ones have finally brought us to a clear understanding of the unit of heredity. The characteristics of sickle cell anemia and most other hereditary traits are defined by changes in protein structure brought about by an alteration in the sequence of amino acids that make up the protein. This sequence in turn is dictated by the order of nucleotides in a particular region of the chromosome. For example, the critical change leading to sickle cell disease is a mutation that replaces a single thymine with an adenine at the position that codes for glutamic acid, converting the position to valine. The sequence of nucleotides that determines the amino acid sequence of a protein is called a gene. Although most genes encode proteins or subunits of proteins, some genes are devoted to the production of special forms of RNA, many of which play important roles in protein synthesis themselves. Synopsis. A half-century of experimentation has made clear that DNA is the molecule responsible for the inheritance of traits, and that this molecule is divided into functional units called genes.